Hello, I'm your narrator, Japing. I will briefly explain protein synthesis as it relates to gel electrophoresis. Protein synthesis is split into transcription and translation. Transcription starts in the nucleus, where DNA is split into single strands using helicase. Then, using a protein called RNA polymerase, this strand is replicated into RNA. This strand replaces diamine with uracil, a base nucleotide part. After DNA is put back together, RNA leaves the nucleus through pores and enters the cytoplasm. Translation begins when RNA binds onto an organelle known as ribosome. Ribosomes read 3-letter codons, and since RNA is single-stranded, transfer RNA carries anticodons to pair up with RNA. With anticodons, tRNA also carries amino acids based on the codons. This forms a chain in the ribosome. Eventually, this chain will fold and shape itself into a protein. That is transcription and translation, key parts of protein synthesis. Now, you will be learning gel electrophoresis. Peace.
Hi, I'm Sean, and I'll be explaining the 4 processes during and leading up to gel electrophoresis. And I will go over each of these 4 steps individually and in detail, starting off with the first step, getting your DNA samples ready and extracting your DNA. To prepare the DNA sample, you must first go through a process called lysing. Lysing is when the cell is broken up using chemical methods, and then the DNA is purified. Now moving on to the second step, which is when we duplicate this DNA strand to have a bigger sample size. This process is known as PCR, or polymerase chain reaction, and it amplifies the DNA into many strands. Cycles, known as thermal cycles, duplicates this DNA each time using something called primers. To simply put it, it just involves enzymes and temperature changes. Now the third step is to cut the DNA strands. Then these cut strands will be used for gel electrophoresis. Using restriction enzymes, DNA is cut as a specific nucleotide sequence. Now to demonstrate this is a strand of DNA. The restriction enzyme used is called a HALE. As you can see, the DNA is cut at specific points based on the sequences. Snip. Now the fourth and final step is obviously gel electrophoresis. Gel electrophoresis starts with preparing an agarose gel. This is poured into a tray and wells are formed later using the DNA strands. Then the ends of the tray will be charged with a positive or negative charge. A type of dye along with the DNA strands are put into the wells. As DNA moves toward the positively charged end, shorter strands move faster and vice versa. The stained DNA can be visualized as bands, and with many bands running down the tray. From there, the results can be compared to match the DNA samples.
We hypothesized that the death of Anna Garcia was as a result of blunt force trauma due to a head-on collision with the stool in the crime scene of the lab. The fall was most likely due to an intake of pills and the previous concussion from her soccer practice. Evidence on the scene shows blood on the stool as well as the floor. This leads us to believe that there was a direct collision with her head in the stool. HIPAA stands for Health Insurance Portability and Accountability Act. This act states that insurance companies and health public agencies can have access to your medical history. For example, public health agencies would need your medical history in case you had HIV or other infectious diseases. Additionally, family and friends in law enforcement agencies cannot have access unless granted permission or they possess a warrant.